Thanks for joining us in another fascinating journey through the world of science here on the SANS Science Experience.
It's great to be diving in again.
If you're finding these deep dives into scientific topics interesting, please do hit that like button and subscribe. Your support really helps us bring you more of this kind of content.
Absolutely. And today we're tackling something really fundamental, dormancy. You can also think of it as life hitting the pause button.
A pause button, yeah. A biological savings account, maybe. A way to conserve energy and just wait out the tough time.
Exactly. From tiny bacteria all the way up to mammals like us. It's this incredible strategy where organisms just dramatically slow everything down.
Sometimes almost to a standstill, it seems. And then, crucially, they can just spring back to life when things get better.
That's the amazing part. We're going to look into how this life on hold actually works, the mechanisms behind it.
How cells, even whole embryos, can just suspend operations and then, remarkably, restart.
Which is what we aim for here, giving you those key insights clearly and efficiently. So let's unpack this world of biological dormancy.
First off, terminology. I notice people use terms like dormancy, quiescence, resting state. Are they all the same thing?
That's a great point. They're often used interchangeably, but there are nuances. Dormancy is probably the broadest term, coming from the Latin for to sleep.
Okay, the big umbrella.
Right. Quiescence, also from Latin meaning to rest, is often used more specifically, especially for cells that have temporarily stepped out of the active cell division cycle. Think of it like exiting the highway onto a rest stop.
Ah, okay. So hibernation is dormancy. A resting cell is often called quiescent. Makes sense.
And even within dormancy, you can have shallow, short-term states like torpor, or really deep, long-term ones like a seed waiting years to germinate.
So despite the different names and depths, are there core rules for what counts as true dormancy or quiescence?
Yes, definitely. There are three really essential criteria. First, it absolutely has to be reversible. The cell or organism needs to be able to wake up.
No waking up, not dormancy. Got it.
Second, its identity must be safeguarded. A dormant muscle stem cell, when it reactivates, should still be a muscle stem cell.
It doesn't change into something else while it's asleep.
Precisely. And third, related to that, it has to maintain its developmental competence. If it's meant to divide or differentiate later, it needs to retain that potential.
That helps distinguish it from, say, cells that are permanently stopped, like our neurons, right? Or those senescent cells that are basically just old and damaged.
Exactly. Terminal differentiated cells have stopped dividing as part of their job description. Senescent cells are often irreversibly shut down and accumulate problems. Dormancy is about this controlled, reversible pause.
So where do we see this playing out in mammals, in us?
It's surprisingly critical. Dormancy, particularly quiescence, is key for maintaining our tissues for regeneration and even in reproduction.
Right, because while most of our cells are those specialized non-dividing types,
We have these reserves of stem and progenitor cells tucked away in tissues.
the backup generators.
That's a good analogy. And a subset of these adult stem cells exists in a dormant or quiescent state within protective niches.
Hiding out, basically.
Sort of. They minimize their activity, their division, their metabolism. This protects them from wear and tear, from stress, preserving their ability to function long-term for tissue repair. We find them in blood, muscle, brain, skin, lots of places.
And you mentioned reproduction, too.
Yes, embryonic diapause. This is fascinating. It's where an early embryo can pause its development before implanting in the uterus.
Wow, why would it do that?
It's often a strategy to time birth for better conditions. Maybe the mother is still nursing a previous litter, or food is scarce. The embryo just waits. It decouples fertilization from implantation and birth.
That's incredibly clever. Okay, let's zoom in on those adult stem cells. What's actually keeping them quiet? What are the molecular controls?
Well, at the heart of it is cell cycle control. These quiescent stem cells are mostly rested in a phase called GOZO. It's like being parked outside the main cycle of division.
So how do they park there? What are the brakes?
The brakes are largely proteins called cyclin-dependent kinase inhibitors, or CDKIs. They counteract the proteins, the CDKs, that normally push the cell cycle forward.
And we hear names like P21, P27. Are they the main players?
They're definitely key ones. P21 often helps initiate the exit into G0. If you lose P21 in some stem cells, they tend to cycle too much and can get exhausted, especially under stress.
Okay, so P21 helps put the brakes on initially. What about P27?
P27 seems really important for maintaining that quiet state, restricting re-entry into the cycle. Loss of P27 often makes stem cells proliferate more, sometimes differentiating when they should be staying quiescent.
So it's like P21 hits the brakes, P27 keeps the parking brake engaged.
Yeah, that's not a bad way to think about it. And there are others, like P57 and the RB family of proteins. They all work together, often in specific ways in different tissues, to enforce that G0 arrest. HES1 is another factor involved, promoting arrest and preventing premature aging.
A whole network of brakes. But it's not just about stopping division, right? You mentioned saving energy.
Exactly. Metabolism is hugely rewired. A major player here is the mTOR pathway. mTOR is like a master center and regulator of site growth and metabolism.
And in quiescent cells, mTOR is generally
dialed way down. It makes sense if the cell's trying to conserve resources. It shouldn't be in a high-growth, high-energy consumption mode. mTOR integrates signals about nutrients, oxygen, growth factors.
So when things are scarce, signals tell mTOR to power down.
Precisely. Pathways involving proteins like PAY10 and the LKB1 AMP-KPAC complex become active under stress, like low nutrients or oxygen, and they put the brakes on mTOR.
Like in the bone marrow niche for blood stem cells, which is low oxygen.
Exactly. In that hypoxic niche, PATO and LKB1 are often upregulated, keeping mTOR suppressed and the hematopoietic stem cells, HSCs, quiescent. If you artificially force mTOR activity, these HSCs can lose their quiescence. We see similar things in neural stem cells and muscle stem cells.
So mTOR is like a dimmer switch for cellular activity, turned down low in dormancy.
A very important dimmer switch, yes. And closely linked to this is the insulin FoxO pathway.
FoxO proteins, I've heard of those, involved in lifespan.
Across many species, yes. When insulin signaling is low, like during nutrient scarcity, FoxO proteins move into the nucleus and turn on genes involved in cell cycle arrest, stress resistance, DNA repair, even autophagy, basically, survival and maintenance programs.
So they actively promote the quiescent protected state?
Very much so. They help activate some of those CDKIs we mentioned, like P27. They boost DNA repair. They help clear out damaged components through autophagy. And they reduce damaging reactive oxygen species, or ROS. Quiescent cells are actually quite sensitive to ROS, so minimizing it is crucial.
Makes sense. If you're in it for the long haul, you need to keep things clean and stable.
Absolutely. Deleting FoxO proteins in HSCs, for example, causes them to exit quiescence and accumulate ROS damage. Activating FoxO can actually induce dormancy in HSCs outside the body. Similar importance is seen in neural and muscle stem cells.
Okay, so we have cell cycle breaks, CDKIs, RB, metabolic downregulation, mTOR suppression, and active maintenance programs, FoxO. Any other major signaling pathways involved?
Notch signaling is another important one, though its role seems a bit more tissue-specific.
How so?
Well, canonical notch signaling doesn't seem essential for keeping HSCs quiescent, but it's critical for maintaining quiescence in muscle stem cells and neural stem cells.
So in muscle stem cells, Notch is active during quiescence.
Yes. If you delete its key mediator, RBPJ, the muscle stem cells leave quiescence, they deplete, and they fail to regenerate properly. There are interesting links, too, like suppressing Notch seems to be part of how activating the PI3K-mTOR pathway pushes muscle stem cells out of quiescence.
And in neural stem cells?
Notch helps maintain their stemness, partly by controlling key genes like SOX2. Loss of Notch signaling leads to differentiation and depletion of the neural stem cell pool.
It's amazing how these pathways intertwine. Now, let's dive deeper into the metabolism. You said it's rewired. How exactly?
Well, actively dividing cells typically rely heavily on glucose and mitochondrial oxidative phosphorylation to generate lots of ATP, but this also produces ROS.
The high energy but potentially damaging route.
Right. Quiescent stem cells often shift their strategy. They tend to favor using fatty acids for fuel through fatty acid oxidation, or FAO, and also rely more on anaerobic glycolysis, even if oxygen is around, plus autophagy to recycle components.
Let's break that down. Glycolysis first, using glucose without the full mitochondrial process. Is that universal in quiescence?
It's actually complicated. In quiescent HSCs, particularly in their low oxygen niche, anaerobic glycolysis seems quite important, regulated by factors like HEF1. It might prime them for rapid activation.
Priming them.
Yeah, some studies suggest these HSCs are sort of metabolically poised. Inhibiting glycolysis doesn't bother the deeply quiescent ones much, but it kills the ones that are primed to start cycling.
Huh. What about other stem cells?
It varies. In neural stem cells, high glycolysis also seems linked to the quiescent state, maybe preparing them for action. But decreasing glycolysis seems necessary for them to actually differentiate. In muscle stem cells, it's different again. Glycolysis seems more linked to proliferation after activation, not quiescence itself. Hair follicle stem cells also seem to use glycolysis to get ready for activation.
So glycolysis isn't a simple on-off switch for quiescence. It depends on the cell type and maybe the depth of quiescence.
That seems to be the case. It highlights the need to look at specific contexts.
What about lysosomes? Those are the cell's recycling centers.
Yeah, there's an interesting idea that lysosomal activity might act like another dimmer switch, controlling the depth of quiescence. Some studies in fibroblasts suggest inhibiting lysosomes leads to a deeper, irreversible arrest, while boosting them leads to shallower quiescence.
And in stem cells.
Quiescent HSCs actually show enlarged lysosomes but lower overall activity. Making them even less active seems to enhance quiescence. In neural stem cells, they also have enlarged lysosomes in quiescence, but the functional role is still a bit debated.
Okay, now fatty acid oxidation, burning fats for energy, is that more consistent?
FAO does seem to be a more common feature, yes. It appears to be a preferred energy source in quiescent cells across several tissues.
Like HSCs.
Definitely.
Yeah.
FAO is significantly increased in quiescent HSCs and seems necessary to maintain the pool. Inhibiting it pushes them to proliferate and deplete. This is regulated by things like PPAR signaling.
And neural stem cells, muscle stem cells.
Same story. Quiescent neural stem cells rely on FAO. Blocking it messes up neurogenesis. Decreasing FAO alone can actually push them out of quiescence. Muscle stem cells also need FAO to stay quiescent. Inhibiting it triggers activation or differentiation.
But is more always better?
Not necessarily. There seems to be an optimal level. Too much FAO can cause oxidative stress. It's about balance. But the general shift towards relying on FAO during quiescence seems pretty consistent. And then when they activate, they switch back to the high-gear mitochondrial metabolism? Largely, yes. Reactivation and proliferation usually involve ramping up mitochondrial activity and oxidative phosphorylation again.
Okay, one more metabolic process. Autophagy, self-eating.
Right. Autophagy is crucial. It breaks down old or damaged parts like mitochondria, that's called mitophagy, which helps suppress ROS. It also generates building blocks and ATP, especially under stress, so it builds resilience and supports the low-energy quiescent state.
So it's like cellular housekeeping and energy generation combined.
Exactly. It's critical for maintaining HSCs, especially under metabolic stress. Deleting key autophagy genes like at G7 or at G12 in HSCs leads to increased ROS, problems with self-renewal, and exit from quiescence.
And in other stem cells.
Similar findings. In muscle stem cells, loss of autophagy leads to senescence, that irreversible arrest because of mitochondrial problems and ROS. Interestingly, autophagy is also needed for them to exit quiescence properly after injury. In neural stem cells, autophagy is boosted during starvation-induced quiescence and seems important for maintaining the pool, likely by clearing damaged organelles and keeping ROS low.
So metabolism is profoundly reshaped. What about control after the genes are transcribed? Post-transcriptional regulation.
Absolutely critical. You need tight control over which proteins are actually made, how many, and how long mRNAs stick around, especially when transitioning into such a low-activity state.
Like controlling the rate of protein synthesis itself.
Yes. Quiescent stem cells generally have very low rates of protein synthesis. They're smaller, less RNA, less protein overall. But again, it's about balance. Too much or too little protein synthesis can impair HSC function, for instance.
How is that controlled? Still mTOR.
mTOR plays a role, yes, through factors like 4-EBP. But other pathways controlling translation initiation, like involving EIF2, are also important. It's finely tuned.
And microRNAs, those tiny RNAs that regulate gene expression.
Huge players. miRNAs can fine-tune the expression of many genes simultaneously. They're critical for quiescence in HSCs, muscle stem cells, epithelial stem cells. For example, miR126 helps keep HSCs quiescent, partly by dampening PI3 CATAC signaling.
Any other example?
In muscle stem cells, miR489 is high in quiescence and suppresses an oncogene called DEC. Another one, miR31, actually binds to the mRNA for a key muscle differentiation factor, mu5. The mRNA is there, ready to go, but miR31 keeps it silenced in granules until the activation signal arrives.
Wow, like keeping the instructions ready but locked away until needed.
Exactly, allows for a rapid response. Then there's mRNA stability itself, controlled by chemical tags like M6A methylation.
N6A.
N6-methyladenosine. It's a modification on RNA that can affect its lifespan, often targeting it for decay. There are writer, eraser, and reader proteins that manage this mark.
And this affects stem cell dormancy.
It seems so. In HSCs, M6A readers help control levels of key regulatory mRNAs like MICAC. In muscle stem cells, an M6A reader called WIFE-DC1 is needed for proper exit from quiescence. An M6A eraser, FPO, is important for neural stem cell function. It's another layer of regulation ensuring the right genes are expressed at the right levels at the right time.
Okay, layers upon layers. What about the packaging of the DNA itself? Epigenetics.
Also crucial. The way DNA is packaged, the epigenetic landscape controls which genes are accessible. Key players here are the polycomb repressive complexes, PRC1 and PRC2.
They add repressive marks to histones, right? Like H3K27E3.
Exactly. H3K27E3 is a mark associated with gene silencing. Quiescence is often associated with increased levels of this mark, helping to suppress genes involved in cell division and differentiation, keeping the cell locked in that stem-like quiescent state.
So removing these complexes or marks would push cells out of quiescence.
Generally, yes, or it messes up their function. For example, deleting PRC2 components in HSCs impairs their self-renewal. In neural stem cells, H3K27E3 helps prevent premature differentiation. In muscle stem cells, PRC2 is needed for proper self-renewal and lineage fidelity. It's about maintaining the right transcriptional program for the quiescent state.
One last thing on adult stem cells. Protecting the genome. Quiescence might protect from replication errors, but damage can still happen, right?
Absolutely. Environmental insults, metabolic byproducts, damage occurs. Quiescent cells need robust DNA repair. They mainly rely on a pathway called non-homologous end joining, NHEJ, because the other main pathway, homologous recombination, requires a sister chromate, which you only have after DNA replication in cycling cells.
And NHEJ is less accurate.
It can be more error-prone, yes. This might contribute to the decline in stem cell function we see with aging, as small errors accumulate over time.
There is also that idea, the immortal strand hypothesis, that stem cells might keep the original DNA template strand.
Karen's hypothesis from 75, yeah. The idea is that during asymmetric division, where one daughter cell remains a stem cell and the other differentiates, the stem cell might preferentially hang on to the older template DNA strand to minimize copying errors passed down the stem line.
Has that been proven?
The evidence is mixed and seems tissue-specific. It's been observed in quiescent muscle stem cells, in some neural stem cells, but maybe not in HSCs or others. It's still an area of active research.
Okay, fascinating. Let's switch gears now to that other major example, embryonic diapause, pausing development itself.
Right, this reversible arrest of the early embryo. Many mammals can do this, delaying implantation. It can be obligate, happening every cycle, like in roe deer, or facultative, triggered by stress, like lactation, or starvation, like in mice.
And it's hormonally regulated?
Yes, primarily by maternal hormones that make the uterus unreceptive to implantation and halt embryonic growth. But we're focusing here on what's happening within the embryo itself.
How do scientists study this? It must be hard to catch in the wild.
It is. Most work uses lab models, especially mice, where diapause can be induced experimentally. And increasingly, researchers use embryonic stem cells, ESCs, derived from that early embryo stage. You can induce diapause-like states in ESCs in culture using inhibitors.
What have these models told us? Are the mechanisms similar to adult stem cell quiescence?
There's remarkable overlap in the regulatory pathways, which is really interesting, but also some differences.
Look, the cell cycle. Do diapause embryos stop dividing like quiescent stem cells?
Not always. Mouse embryos in diapause pretty much halt division, with CDK inhibitors like P21 being upregulated, similar to adult stem cells. But roe deer embryos in diapause actually continue to proliferate, just very slowly.
Huh, interesting difference.
And even in the mouse models using ESCs, inhibiting mTOR slows the cell cycle dramatically, but doesn't always cause a complete GOG01 arrest like you see in the in vivo embryo or with other inhibitors. So there are subtleties.
What about those core pathways? mTOR.
Definitely downregulated. The whole PI3 kinselin mTOR axis is suppressed in diapause mouse embryos. And importantly, inhibiting mTOR pharmacologically is a very effective way to induce a stable, reversible diapause-like state in cultured mouse embryos and ESCs.
So mTOR inhibition is a key trigger or maintainer of diapause, just like in adult quiescence.
It seems to be a central hub. And upstream regulators like the LKB1 AMP pathway, activated by starvation, also play a role in inducing this pause state in ESCs by inhibiting mTOR, similar again to HSCs. Amino acids activating mTORC1 are needed for reactivation in roe deer.
And the FoxO pathway.
Also essential, at least in mice. FoxO proteins are active during diapause. They seem particularly important for shifting metabolism towards using lipids, which are a preferred energy source during diapause. Inhibiting FoxO1 compromises embryo survival. Upregulation of FoxO is also seen in diapause in other species like mink and tamer wallabies.
So cell cycle breaks, mTOR down, FoxO up. The core regulators seem conserved between adult quiescence and embryonic diapause. What about the detailed metabolism?
A very similar profile again. Decreased mitochondrial oxidative phosphorylation, increased reliance on lipid metabolism, and increased autophagy. Reactivation involves ramping up mitochondrial activity.
Is glycolysis complicated here too?
Yes, there are some conflicting reports actually. Some studies show low activity. Others suggest glycolysis might still be important, perhaps for maintaining readiness or specific biosynthetic pathways even during the pause. More research is needed to clarify its exact role during diapause itself versus reactivation.
But the shift to using fats, FAO, is clear.
That seems much clearer. Diapause mouse embryos accumulate lipid droplets, and removing them impairs survival. Boosting FAO can even enhance survival in culture. A factor called TFCP2L1, important for pluripotency, actually promotes FAO, and embryos lacking a struggle to enter diapause properly when mTOR is inhibited.
Are there pathways unique to embryonic diapause, related to maintaining the embryo's unique state?
Yes. Signaling pathways that maintain pluripotency, the ability to form all cell types seem crucial during diapause. Lifstat signaling and WANT signaling help reinforce these pluripotency programs in the epiblast cells, the ones that form the future embryo proper.
Why would they be needed during a pause?
It might be that maintaining this unique potent state over a prolonged period requires continuous reinforcement from these pathways, even when overall activity is low. It might explain why these factors are needed to culture ESCs long-term in the lab, even though they aren't strictly essential for normal, brief pre-implantation development.
Interesting. Any overlap in the post-transcriptional or epigenetic controls?
Definitely. mRNAs are involved. For example, LET7, a mRNA known to regulate developmental timing, is upregulated in diapause and can suppress mTOR and myloc. There's even evidence it might be delivered from the mother via extracellular vesicles to help trigger diapause.
Maternal control through mRNAs, wow.
And that M6A RNA modification we talked about. It also plays a role in controlling key mRNAs like MING during diapause. Epigenetically, while the cells maintain their pluripotency programs, the overall reduced activity leads to some changes, like depletion of certain activating histone marks and accumulation of repressive marks and DNA methylation, along with condensed nucleoli indicating reduced ribosome production.
So, summing up this amazing phenomenon of dormancy, what are the big takeaways and where does research go next?
Well, I think three things stand out. First, entering and exiting dormancy isn't just a passive shutdown, it's an active, tightly regulated process. Second, there are clearly common molecular themes like the roles of mTOR, FoxO, metabolic shifts towards FAO, autophagy that pop up across different types of dormancy in different organisms.
But the third point is
that there are also crucial tissue-specific and species-specific differences. The exact role of glycolysis or notch signaling, for instance, isn't universal. We need to consider the context.
Right. Most dormant states seem to reduce overall anabolism protein synthesis, high-energy metabolism, but the specifics vary.
Exactly. Most knowledge so far comes from comparing dormant versus active cells at static time points. What we really need now are more dynamic studies. How exactly does the transition into dormancy happen over time? What are the very first triggers for exit? How do these complex networks interact temporally?
Trying to separate the drivers from the passengers in these signaling networks.
Precisely. And really understanding reversibility is key. What stops a dormant cell from tipping over into irreversible senescence? Identifying those factors could be huge, maybe helping us find ways to reactivate dormant stem cells for therapy.
And the implications for disease. You mentioned cancer earlier.
Yes, dormant cancer cells are a major challenge in treatment, as they can survive therapies and cause relapse later. Understanding the fundamental biology of dormancy could provide new strategies to target these persistent cells.
It's truly a fundamental aspect of life, this ability to pause and resume. So many potential applications from regenerative medicine to oncology.
Absolutely. A remarkable testament to life's adaptability.
As always, don't forget to like the video and subscribe if you haven't joined us yet.
Your support keeps these explorations going.
And maybe take a moment to consider this incredible power of the pause button in biology, how life waits, adapts, and endures.